All right, well, welcome back, everyone. So we are going to continue here, looking at the innervation of our heart, the stimulation of our heart that leads to a contraction. So first thing that we'll pick up here, talking about, are some of the features of the heart in terms of the different types of cells that we have that actually contribute or that lead to the actual contraction, right? So we talked about earlier that there's the cardiac myocytes, right? So the actual muscle cells that are actually contracting and leading to the squeezing of the heart and the ejecting of blood. You also have the conductile cells, if you will, right? So the cells that are part of this conduction system here that we're going to discuss. So if you recall from our last part, we were talking about the conduction system, and we started at the SA node and we went down to the AV node. We'll kind of review some of that and talk about that here in a little bit. So this basic conduction system can be kind of summarized in this slide here. So we're going to start in the SA node. Again, that's the pacemaker, right? And we'll talk about something called the pacemaker potential here in just a little bit. But we're starting in the SA node. Remember, it's in kind of the posterior upper wall of the right atrium. That cell, for reasons that we'll talk about in a moment, is going to generate an electrical impulse. It's going to spread across the atria, and then it's going to reconvene back to the AV node, and then it'll spread down to the bundle of His, to the AV, the bundle branches, to the Purkinje fibers, and so on, right? So again, some of the stuff that we talked about previously, right? So that's the spreading of the signal, right? And we'll talk about that more here. That'll be kind of the focus of this part. And then you actually get the sending of that electrical signal to the muscle cells, right? So to the actual cardiac myocytes that then respond by contracting, right? The mechanism by which the cardiac muscle cells contract, it's essentially the same as how our skeletal muscle contracts, right? So it's striated, you have the sarcomeres, you have overlapping actin and myosin, you have to have calcium, it binds to the troponin, that shifts the troponin-tropomyosin complex, right? The myosin head binds to the actin, so on and so on, right? So it's the same basic process as that. So if you need a review on how the muscle is going to contract, go back and watch some videos on the skeletal muscle contraction, right? Because it's essentially going to be the same general type of process, right? So what we're going to focus on in this part is not necessarily these cardiac muscle cells and the signal that's going on there, but we're going to focus on the actual conduction, right? And so what's happening to initiate that signal in those SA nodal cells, right? So again, the SA nodal cells, these are the ones that are initiating the contraction, right? So again, you'll hear them called the pacemaker. Those SA nodal cells, they have a baseline membrane potential like all other cells, and it's about -60 millivolts, right? So again, that's always inside of the cell relative to the outside of the cell, right? So it's about -60 millivolts, right? Now, this membrane potential, sometimes you may see it called the resting membrane potential. However, some texts or some books may not use that term because it's not resting in the sense that it's not steady, right? It's not at this constant level, but it can change, right? And that change is what's contributing to us, what's contributing to our heart's ability to kind of spontaneously initiate this signal, right? So you may also see it called the pacemaker potential, right? Because it's not steady, it's not at rest, it's always kind of changing and fluctuating, and again, that's contributing to that automaticity, autorhythmic function or feature of the heart, right? So the process by which this happens, it's going to involve sodium, it's going to involve potassium, it's going to involve calcium, right? So if you recall from our action potential discussions when we talked about it with the neuron, the two main ions that we were discussing there were sodium and potassium, right? So we'll see those, but we're going to add the third one. We're going to add calcium, and calcium has a very important role to play in our heart's physiology, right? Not only just for the initiation of the signal in these conducting cells, but also in the actual cardiac myocytes as well. Calcium is going to play a really big role there as well, right? So here again, you can see those SA nodal cells, again, in the SA node, the negative 60 resting membrane potential or the pacemaker potential. And notice here that we have our different channels, right? So we've got our sodium channels, we've got maybe potassium channels, calcium channels, and so on, right? And so on. So the channels that we're going to talk about are going to be initially those that are involved with sodium. We'll then move to calcium, and then we'll talk about the role of potassium in this initiation of our electrical signal, right? So these are three kind of main phases that we're going to talk about, and again, you can see those relevant ions. So sodium first, then we'll talk about calcium, and then we'll talk about potassium. So the three main phases, we're going to start at that membrane potential, right? We're going to work our way up to threshold. Again, threshold should be a term that's familiar from our action potential discussions, right? So we're going to work our way up to threshold. We're then going to get depolarization, and then we're going to get repolarization, right? Now, one just kind of little piece of information I'll tell you about this whole process, and then we'll get into the details, is that for the, and again, this is specific to our, what we're going to call the pacemaker potential in the SA node. Each time the SA node initiates a signal, right, that corresponds to one heartbeat, right? One heart contraction, one contraction of the heart, right? So what we'll see is that if you alter the rate at which you get these pacemaker potentials, then subsequently you're altering the rate of the heart, right? You're altering the heart rate, how many times it beats per minute, right? So typically we get about one of these pacemaker potentials, one of these signals that's initiated about every 0.8 of a second, right? So it starts about 0.8 of a second from the previous one. So about every 0.8 of a second. That corresponds to about 75. However, our SA node on its own, okay, our heart rate on its own would be much higher than it actually is. Okay, so our typical heart rate, excuse me, our typical heart rate is, maybe somewhere between 60 and 100. Okay, it varies depending on lots of different factors. Okay, if we didn't have our nervous system, it would be about 100. Okay, that would be the relative baseline. Okay, for a heart rate is 100. That's faster. That's higher than what most people's typical heart rate is, resting heart rate, things like that. And the reason it's not 100, right? The reason that it's much lower, maybe 75, some people 60, some people 50, it just depends, is because of something called vagal tone, right? Vagal tone. So that first word there, vagal, right? That should look familiar. That should look like vagus, right? And so this is referring to the vagus nerve. So what vagal tone is it's essentially the innervation of the heart via the vagus nerve that keeps the kind of overall heart rate that we actually get in the body, it actually keeps it a little bit lower than what we would have otherwise, right? And that should make sense because the role of the vagus nerve, right, it's a parasympathetic, it has a parasympathetic function. It's helping to decrease heart rate, right? That's the parasympathetic effect on the heart, right? So again, normally, if we didn't have that innervation, if we didn't have that input from the autonomic nervous system, the vagus nerve, it may be a little bit higher, but because we do have it, it's going to be a little bit lower, right? So maybe somewhere around 75 as an average, right? So if we look here, this is the actual activity that's going on in the SA node, right? In those SA nodal cells, right? And we can divide this into these three main phases, right? So we'll have our membrane potential up to threshold, we'll have the depolarization phase, and then we'll have the repolarization phase, right? So this first phase is, again, depending on what text you look at, it may be called something different. Here in this image, they're calling it pre-potential, okay, pre-potential. In this image here, they're calling it pacemaker potential, right? The specific name that you give to it doesn't matter a whole lot, so long as you know what's happening and what are the events that are occurring here, right? So we'll call it pre-potential, we'll call it pacemaker potential, right? So what's happening is you're starting off at that baseline, right? You're starting off at that resting potential, that membrane potential of negative 60 millivolts. Again, remember that's inside relative to the outside, negative 60. And you have sodium gradually begin to come in. And this is constant. Okay, so sodium in the SA nodal cells is constantly and routinely flowing into the SA nodal cells because of these sodium channels, right? Because of these sodium channels. And you may see them sometimes they're called funny current channels, right? They're called funny because they're actually permeable to sodium and potassium, right? But that's mostly beside the point. So you have this orange phase here. Again, you've got this rising phase as we reach up to threshold, maybe somewhere around negative 40. Okay, that's the threshold. And when you get that, toward the end of this pacemaker potential, at the end of this pre-potential, kind of this latter half here, you then also begin to have some calcium come in as well, right? You then reach threshold, all right, and then the depolarization phase, again, in our SA nodal cells, is essentially all calcium, right? So you get this massive influx of calcium due to voltage-gated calcium channels opening, and calcium is then rushing into the cell, right? So whenever calcium is rushing into the cell, it's making the membrane potential of those SA nodal cells, it's making it increase, right? So we're continuing to move in the positive direction, right? Now, this is a main difference between this potential, this pacemaker potential process that we're discussing here, and our neuronal action potential we've discussed in previous lectures and courses, right? In the neuronal action potential, the depolarization phase, it wasn't calcium, it was some other ion, right? Do you remember what that other ion was? It was sodium, right? So in the depolarization phase of our neuronal action potential, it was sodium. Here, it's calcium, right? So that's going to be a difference between this and our neuronal action potential, right? So we've got calcium rushing in, and then we're going to go all the way up to roughly positive 20 millivolts or so, and those calcium channels are then going to close, right? So we're going to close our calcium channels, and then we're going to have potassium channels open, right? Remember, potassium is highly concentrated on the inside of the cell. You open it up, it's going to move down its electrochemical gradient, it's going to move out of the cell, and it's going to bring the membrane potential back down, right? So we have this efflux of calcium, of potassium. This is very similar to what you have in the neuronal action potential, right? The repolarization phase in the neuronal action potential, potassium efflux, right? We have the same thing here, potassium efflux. Potassium is leaving the cell, bringing our membrane potential back down about to negative 60, and then the process will start over, right? So all of that there, right? So from the beginning of this orange line here to the end of this blue line here, that corresponds to one heart contraction, right? One heart contraction, right? So the heart beat one time, right? So you can imagine what's going to happen if you stretch that out or if you push it together, right? You're going to, in some way, shape, or form, you're going to alter the rate at which the heart is contracting, right? And we'll talk about how that happens and why that happens and why we may want that and why we may not want that. We'll talk about that in later parts, right? So again, we can see this kind of, we can put all this together and we can see this total process, again, even including what we talked about in our last part, right? So you start the signal in the SA node, that's this signal here, right? We're starting this pacemaker potential process, right, in our SA node, right? Starts in the SA node, spreads across the atria, right, via those gap junctions, comes back to the AV node, so it comes back here to our AV node, and it's delayed, right? So why the delay, right? I think we mentioned this in one of our last parts, or at least mentioned that you have a delay, but not maybe why you have it. So you have the delay primarily to allow the signal to stop for just a brief moment to allow the ventricles time to fill, right? So to give the ventricles enough time to fill with blood before the ventricles then contract, right? So you can imagine what would happen if as soon as the atria contracted, right? And remember, some of the blood that goes into the atria, as we'll see later, so some of the blood that's leaving the atria and into the ventricle, it gets there because of the atria contracting. Some of it gets there just because of gravity. Some of it gets there because of the atria contract. So you can imagine what would happen if there was no delay, and as soon as the atria contracted, the ventricles then immediately contracted afterwards, right? What that would mean is that as blood is then coming down into the ventricle from the atria, the ventricle is also contracting and pushing blood up out of the heart, right? And some of that may result in blood going backwards into the atria. It may result in this kind of turbulent mixing of blood. But the idea here is that because you get this delay, you have time for the ventricles to fully fill, right, or mostly fill, and then the signal is reinitiated, right? So the signal reinitiated, it leaves the AV node, goes down the AV bundle or bundle of His to the bundle branches, into the Purkinje fibers, and then into that ventricular myocardium, and it gets the ventricular muscle to contract and then eject the blood up and out, right? But you have to have that delay, right? So again, you go SA node to AV node to that AV bundle or bundle of His to the bundle branches, and then to the Purkinje fibers, right? And again, you get this very quick initiation. You get this very quick signal moving down, especially with the Purkinje fibers. The Purkinje fibers are really large, so they allow that nervous impulse to travel very, very rapidly, and it allows the ventricles to contract in sync with this what word am I looking here? Looking for synchronous, right? So you get this synchronous contraction of the ventricles as the signal is flowing down in the Purkinje fibers. Ventricles contract, and then you're ejecting all this blood up and out, right? Up and out. So, and again, all of this signal or this process that we're talking about, this was specific to our SA node, right? So to the pacemaker potential cells, right? So this process that we're looking at here, right? That signal that initiates these signals or these pathways here, right? So you have to start here. You have to start with this pacemaker potential, and then you can go from the SA node to the AV, gets delayed, moves down into the bundle of His, bundle branches, Purkinje fibers, and so on, right? Now you can also have what are called ectopic pacemakers, right? And so ectopic meaning outside, right? So in this case, outside the heart, other than the SA node, right? And so you can have other cells that can also kind of act as a pacemaker, and some of these result in a faster heart contraction. Some of them result in a slower heart contraction. It just kind of depends on which specific set in that pathway is initiated, right? So many of these are depolarizing slower than the SA node. So remember, baseline our heart rate is maybe somewhere around 75, right? And so you could have the AV node be the default pacemaker if something is wrong with the SA node, and this gets reflected in our ECG or our EKG, as we'll see. So in this case, the baseline, if it was the AV node or the atrioventricular node, is 40 to 50. Okay, this is fast enough to sustain life. Many people's heart rate, especially if you are very physically fit, you have good aerobic endurance, oftentimes it can be in the 50s, 40s, in some cases really, really low 40s, right? So that's sufficient enough to sustain life. If it's the heart muscle itself that's initiating the signal, 20 to 40 beats per minute, that's difficult in terms of being able to eject an appropriate amount of blood each time to perfuse your tissues, to deliver oxygen, and to sustain life. So that in that case, it may be not ideal, right? But the SA node, it's the fastest pacemaker that we have in the heart. What we'll see later when we look at our EKG, if the signal is not coming from the SA node, then you get a different waveform, right? And so what we'll see is that you have this kind of alternative shape to the ECG when the signal is not being initiated from our SA node, right? So let's stop here. We'll then pick up in our next part, and we'll begin looking at cardiac myocytes and we'll begin looking at the actual electrical process or electrical flow that's coming through our muscle cells that occurs after the process that we just discussed.